Good morning, good afternoon, good evening to wherever you are when you're watching this. My name is Rob Schick, and I'm going to be talking about a new case study that we're spinning up. And that case study is called The Population Consequences of Multiple Stressors. And I want to take a minute to acknowledge all my co-authors that are contributing to this case study, and they are listed below. So there are two primary questions we're going to be looking at during this multi-year research effort, and they are: what are the links between multiple stressors, health, and vital rates in individual right whales? And two, which of these stressors can be reduced in practical ways to lower extinction risk for the animals? So there's been a lot of work done to figure out, what is the impact of a single stressor on a right whale. And what we're trying to do in this project is understand that and extend that so we can look at how they may interact with other stressors. So we have this idea of what the cumulative impact of multiple stressors may be on these individuals and on this species. The work is being funded by CERTIP and the U.S. Office of Naval Research. The lead institution on this whole project, the PCOMS project, is University of St. Andrews, where Peter Tyack is the PI. And therein, there are going to be two primary case studies. We'll be looking at bottlenose dolphins in Barataria Bay and North Atlantic right whales, which will be the focus of my talk today. It bears repeating just because it drives a lot of our work, and by our, I mean the whole entire community. This is a critically endangered species, and the population is shrinking. We know that individuals experience multiple stressors throughout their habitat range, including, for example, entanglement with fishing gear and ship strikes and noise pollution. Of course, there are many others as well. We have indices of how these stressors can show up in the health of the animals. These can be acute indices like stress hormones, and they can also be chronic. So, an animal that's had a sublethal ship strike and has a scar or a wound, or an animal that has gear trauma and or is carrying gear, like we see in the picture at the upper right hand of the slide. So the impact of these multiple stressors is both important and also unknown, and that's what we're trying to get at in this work here. This is a shot from Enrico Perotta's manuscript in 2018, showing the sort of latest representation of the PCOD model, which used to, of course, be called PCAD. And the idea being that we have stressors or exposure to stressors on the left-hand side that can cascade through this diagram, resulting in changes in the physiology and behavior of animals, then of course the health, and then ultimately the link to the vital rates at both the level of the individual and then the population. So ours is going to be slightly different, as sort of a subset of that. Again, we have these sort of underlying drivers that may be changing: climate change, North Atlantic oscillation, etc. And then we have these multiple stressors. A few of them are listed below. Both of these can result in physiological changes of the animals, which can in turn impact the health of the animals, and then finally can impact the vital rates. And so what we want to do is quantify these arrows that are linking any one of these boxes in this diagram. Our proposed plan is as follows. This is a 4.5-year grant, and in years 1 and 2, we're going to be doing a lot of data collation with a primary focus on Cape Cod Bay, and we'll be gathering things like the individual life histories, health and stressor effects, including information on entanglement and stressors such as prey or noise. And in particular, with respect to prey, I mean sort of prey limitation. We'll be doing a lot of modeling throughout. I'll come on to this later in a couple of slides, but basically taking some of the years and in some cases decades worth of information on prey and noise in Cape Cod Bay and turning these into covariates that can be used in the model that links health to vital rates. And finally, towards the end, we'll be doing field work in Cape Cod Bay, and with the acknowledgement that, of course, that later on in this, things may look a little bit different. And our hypotheses are as follows: that prey limitation may amplify the consequences of poor health induced by exposure to other stressors such as entanglement. Chronic exposure to things like noise in Cape Cod Bay may increase the allostatic load and therefore amplify the declines in health that we see. So again, trying to sort of tease our understanding of what it means for an animal that is experiencing multiple of these stressors, how that actually can translate to our understanding of health and vital rates. The first part of this will be data collation. So we'll be bringing together all of the visual health data that's mostly taken from shipboard, but also from planes. And this work has been pioneered by Heather Pettis and Roz Rolland from the New England Aquarium. And most people are probably familiar with slides like this, but the idea, this is the same animal seen in quite good condition on the right-hand side of the slide, and then quite poor condition, obviously, on the left-hand side of the slide. This is a very, very rich data set dating back many decades and comprising almost 85,000 sightings of whales. 760 individual whales are in the data set. Of course, not all of those are still alive. Similarly, from photographs, we can get information on the impacts of entanglement. So I'm showing you three levels of entanglement injury: minor, moderate, and severe on the animal. And work that Amy Knowlton and I have been doing has shown how the changing severity or increasing severity is resulting in poor health, shown on the graphic on the lower right-hand side here. And this is one, but we want to see like, okay, well, how does entanglement impact health given that animals may also be facing, for example, a poor or rich prey year? So one way we'll do that is to build up the prey database that Stormy Mayo and colleagues have been collecting for decades now. So shown on the right-hand side of this slide is a spatial surface or raster representing the probability of a right whale feeding in Cape Cod Bay on a particular day, in this case February 24th of 2018. On the left-hand side of this is actually where the whales were seen on that particular day. And so one of the things we're going to be doing is building a spatial model of this prey database over all of the years and decades that it's been collected so that we can feed into the PCOD model. Another type of data that we'll be collecting, or have been collected that we'll be collating, excuse me, are the sound data. And these are some snapshot of the data that have been collected in Cape Cod Bay. So this is showing you the yearly specific deployment location of these MARWs in Cape Cod Bay. There are additional years from this farther back in time, and they're slightly sparser the farther back in time you go. But again, the idea is to build this into a covariate that represents how whales are experiencing sound in Cape Cod Bay. In years 3 and 4, again, asterisk with the possibility that the bay may have changed by that time, we'll be collecting all these sorts of data. So we're going to be adding to the drone data that have already been collected in the bay, and there will be ongoing prey sampling that's done by Center for Coastal Studies, as well as the sound recordings that are being done by Cornell. So the aerial photogrammetry data from the drones is great because it allows you, these really, really high resolution shots of the animals that can document change over time. For in this case, I'm showing you the same animal over 4 different years, but it can also show within season changes. And so that's really, really a power of this database. Of course, the within season changes are harder because you need to recite the same animal multiple times, but those data are being added to all the time. So looking at changes over time, both across years and within a season, and also quantifying impact of events like entanglement or a life history cycle, like looking at an animal's change through lactation, as is depicted in these 4 shots. We also hope to use the drone data to help calibrate and refine the VHA data that are taken either from ships or from planes. We'll also be analyzing and doing collection of microbiome data of right whales. And so there's a lot of information on this slide, and I'll sort of gloss through a little bit of it. But the idea is that each one of these dots represents a microbiome community of an individual right whale, and the ellipses sort of show the variance around that by either species or also by area within some species. And what's known about the relationship between microbiomes and the health of individual right whales is not clear, and that's one thing we want to be trying to answer. But in other mammals, there have been links that are clear between, or links that are being shown between health of microbiomes and health of individuals. And so what we want to do with the collected data we have so far is relate some of these scores to body condition, life history, and entanglement events to see how these differences in this multidimensional space that is shown may link to the photographic evidence that we have. And so just one thing to highlight here is that the North Atlantic right whales and the southern right whales seem to be different in terms of their microbiome communities, and that's interesting, of course, because the health of these two species or individuals in these two species is quite different. We'll be doing a lot of modeling throughout this. I mentioned a couple times now how we'll be building up the time series of prey and noise in Cape Cod Bay. We'll also be adding to and hopefully refining work that we published earlier in 2013. So that was one of the original PCOD models. And of course, a lot has changed in the right whale distribution and abundance since that time. And so the spatial component of that model is going to be different, but the flavor of it will be similar. We'll also be doing some dynamic state variable modeling to build and examine future scenarios with respect to disturbance and vital rates. And a good example of that is a paper that Enrico recently published on blue whales, I believe. So cycling back here, again, this is work that Amy Nolt and I have been working on for a while now, and the idea here is that this is a single stressor. So this is survival. What I'm showing you here is survival of male and female right whales as a function of entanglement severity. So this is showing you that for this single stressor, entanglements are bad for whales, and they're worse for females than they are for males. And so that's fine. It's of course terrible for the whales, but it's fine. And what we want to get at in this case study here is how can we extend this so that we're not just understanding a single stressor, but we're understanding multiple stressors. And then I'm linking back to the question that I put at the start of the talk. So we also want to be able to see how these interactions have changed between the stressors and changed over time. Again, we want to be figuring out if there are any that we can isolate and reduce so we can lower extinction risk. And then I mentioned several times that we're focusing on Cape Cod Bay, and that's one of the best and most well-studied areas for right whales, both in terms of its prey and also the whales that are using the area. And so one of the big questions is, well, how representative is that for the broader ecosystem in which right whales live? And then finally, if we can use this information to say, are there any specific types of data that we need to be collecting going forward to monitor this population, then that's what we'll be doing. And with that, I'll say thank you and look forward to seeing you virtually in the consortium.